Welcome to part 2 of the two-part lecture series dealing specifically with DNA damage and the repair of that damage. The first lecture was one dealing specifically with how mutations can occur, or different ways that mutations can potentially arise. And in this lecture, we're going to focus on a handful of repair mechanisms that have evolved in such a way to help maintain the integrity of the DNA. The first example is a mechanism known as mismatch repair, or MMR. This particular mechanism has a series of enzymes involved that scan the genome for mismatches. These are specifically problems that occur with the incorrect nucleotide being added in after or during replication. The first enzyme that's important in this process is one known as DAM methylase. DAM methylase. This is an enzyme that adds methyl groups to adenines prior to replication. Basically, what this does is tags the DNA so that the original parental strands are now marked with the methyl groups. And again, this occurs prior to replication. Once replication has occurred, you end up with two double-stranded DNA molecules. However, with each double-stranded DNA molecule, you have one strand that is methylated or tagged to indicate that that's the original sequence information, as well as the newly synthesized DNA in this particular figure represented by the orange strand. In addition, on this slide, you see an enzyme known as MutS. MutS is an enzyme that occurs as a dimer, meaning that there's two copies to make the functional repair protein. It has an overall horseshoe shape, and its job is to scan the DNA looking for mismatches that have occurred. Additionally, not listed on this particular slide, but we'll see on the next slide, is an enzyme known as MutH, and MutH recognizes hemimethylated sites. Hemimethylated is like what we see in the left-hand figure, where one strand is methylated, representing the original parental tagged strand, and then the other strand has no methyl groups, where the newly synthesized DNA occurs. So MutH recognizes hemimethylated sites, like you see on the figure here, recognizes sites where there's a methyl group on one strand and not on the other. In this slide, we can take a little closer look at what's happening with MutS. MutS scans the DNA for mismatches. Mismatches would be where there's physical distortions that have occurred because an incorrect nucleotide has been added in. The mismatch is recognized, and additional enzymes known as MutL and the original MutH that I mentioned in the last slide are recruited to the target site. These are accessory proteins in this repair process. In particular, MutH is an enzyme known as an endonuclease. It is a specific type of enzyme that can make cuts in the DNA. Additional enzymes are recruited to the target location, including a helicase known as UVRD, as well as an additional exonuclease to help cut out damaged or mismatched DNA. The result that occurs with the recruitment of these enzymes is that a single strand gap has been generated, and hopefully you can see that on the right-hand panel where a stretch of DNA has been removed by the enzymes in question. These enzymes now are removed, and there is a gap that DNA polymerase III can come in and fill, and ligase will close in that last covalent phosphodiester bond. So essentially what's happening here in this mechanism, to summarize it, is a mismatch is recognized, then a piece of the newly synthesized DNA is cut out where the mismatch occurs. The gap that's left behind is filled in with new DNA, and hopefully this time no mismatch will take place. This next repair mechanism that I want to highlight is what's referred to as direct reversal of DNA damage. This is also a mechanism that occurs universally, meaning it's found in both prokaryotes and eukaryotes. What happens in this mechanism? Actually, let's take a step back. This particular mechanism is known as photoreactivation, and it involves fixing thymine-thymine dimers that may have been formed due to UV light exposure. So a few slides back, we discussed the fact that UV light can in fact cause these thymine-thymine dimers to form, where covalent linkages occur between neighboring thymines. This causes a physical distortion in the DNA, and that is recognized by an enzyme known as DNA photolyase, and you can see that in the left-hand panel. This enzyme recognizes the distortion, and it uses the energy from visible light to break those covalent bonds. Hence, the thymine-thymine dimer is removed in the sense that there's no more dimer, there's no more covalent bond between the neighboring thymines, and the DNA is no longer distorted. The repair of alkylation is also a direct reversal of DNA damage. That means there is an enzyme that specifically recognizes these alkylated bases. In this particular case, it is a type of methyltransferase that recognizes these methyl groups on the bases and then removes the methyl groups from the bases overall. So it's a pretty straightforward process. The enzyme recognizes the problem that has occurred and removes the methyl group all in one full swoop. But again, this is considered direct reversal of DNA damage, similar to what we just talked about with photoreactivation. The last two examples are types of excision repair, but they are subtly different from one another. So the example that you see on this particular slide is known as base excision repair, also known as BER. In this particular example, there are additional enzymes that are involved in this particular repair process. There are enzymes known as DNA glycosylases. These are enzymes that recognize incorrect bases and remove by hydrolyzing the glycosidic bond. Essentially, what happens if you follow the sequence of steps in this particular figure through, what you can see in this example is that for some reason there is a base that contains a U in this double-stranded DNA molecule. Now, U's are not supposed to be found in DNA, hence this is recognized as a problem by one of these specific DNA glycosylases. The problem is recognized, and specifically the base itself is cut out. So not the entire nucleotide, but just the base. Once the base has been removed, this is now recognized by other enzymes as a physical distortion because now there is no base that allows for hydrogen bonding between the two strands at that particular point. There is an enzyme known as AP endonuclease that comes in and makes a cut now to remove the remaining portion of the nucleotide. Now there is an actual gap in the sequence where an entire nucleotide is missing. This is recognized by DNA polymerase, typically DNA polymerase 1 if we're talking about prokaryotes, fills in the gap, and then ligase closes that covalent linkage that needs to be closed. This is just another view of base excision repair, and I like this particular figure because it shows you the molecular structure of the nucleotides and how the damage is actually recognized and the base removed overall. So again, same mechanism as we just talked about in the last slide, but this one has more detailed molecular structure so that you can see what is actually taking place. This last example is also a type of excision repair. However, this is referred to as nucleotide excision repair. In this case, damaged DNA is recognized by enzymes that are part of the UVR gene complex. They are UVR gene products. With this specific example, we're noting a thymine-thymine dimer, and that is recognized by a number of enzymes in the process. The enzymes recognize the distortion and make a cut on either side of the problem. So as you can see where the thymine-thymine dimer is formed right at the center of the slide, there's a cut made on either side, and that piece that has the thymine-thymine dimer is removed. Now there is a gap in DNA sequence that can then be filled in with DNA polymerase I and the final covalent linkage sealed up with DNA ligase. Again, there are multiple examples of repair mechanisms. What I wanted to do was highlight just a few for you so that you get a feel for the fact that there are plenty of mechanisms that have evolved to help fix DNA damage. Again, this is just a small snippet. So what we've dealt with to this point, I think, are pretty straightforward types of repair mechanisms dealing with relatively simple or straightforward types of problems. But now I want to transition a little bit into a situation where you might have a little more extensive DNA damage. In this case, this is what's referred to as a double-stranded break. A double-stranded break is exactly what the name implies, in that the two strands of the DNA have been broken, and this definitely needs to be fixed in order to maintain the integrity of the DNA. There are, in fact, two different ways that we'll talk about fixing double-stranded breaks or double-strand break repair mechanisms. The first one that you see on this particular slide is something that is called non-homologous end joining, or NHEJ. This is evolutionarily conserved, meaning that it is found in both prokaryotes and in eukaryotes. In this particular mechanism, broken ends are directly ligated without the need for a homologous template. So in this particular type of situation, the ends of the chromosomes are basically being pasted back together after the damage has broke them into two. However, there are microhomologies that are present in single-strand overhangs that allow for this interaction to take place. So it's not just piecing together random pieces of DNA. There is some sequence homology that allows for this to take place. Now, as you can see from this figure, it is a pretty complex mechanism that involves a number of different enzymes. The first enzyme set that I want to talk about is known as Ku, and it's actually a heterodimer, meaning that it has two functional components that are slightly distinct from one another. Those two components are known as Ku70 and Ku80. These two molecules are thought to potentially have ATPase activity, which means that they can harness the energy associated with ATP, as well as potentially having helicase activity. And we know helicase's job is to unwind DNA. The next enzyme on the list is XRCC4, which is actually a cofactor associated with ligase. Ligase is listed at the end of this particular list of enzymes, and we know that ligase is the enzyme that links covalent bonds to make a seamless piece of DNA. So this XRCC4 plays a role in helping ligase get its job done. DNA PKCS is a DNA-dependent protein kinase, and protein kinases are enzymes that transfer phosphate groups from ATP to a molecule to then typically make that molecule active. And then the last one on this list is Artemis, and Artemis is an enzyme that is a nuclease. So what you can see in the series of steps that takes place is that you have the ends of the molecule recognized by Ku. There is then binding of the protein kinase, which then phosphorylates several components of this enzyme complex. That phosphorylation typically allows for a conformation change, and what you can see in the figure is that the ends of the molecule are coming closer together. There is an alignment step and then final resolution that occurs. And so this is now, again, a way of fixing this break in the double-stranded molecule. The second of the two double-strand break repair mechanisms is known as recombination repair or homologous, or utilizes homologous recombination, also known as homology-directed repair or HDR. In this particular type of repair mechanism, there are identical sequences used as a template to repair the break. It is a similar process like we discussed in the last repair mechanism. However, again, recombination, genetic recombination, is utilized to fix the problem in this particular case rather than just piecing the ends together. There are a number of enzymes involved in this particular process as well, and I'll list some of them in just a second. Basically, what I want to do is just give you the overview of what's happening in this repair mechanism. There is an alignment of homologous molecules. There is processing of the overhangs in the DNA that occurs. There are what's known as strand invasion steps, which are key components to genetic recombination. And then there's new DNA synthesis that occurs. So, enzymes that are involved in this particular process, there is an enzyme known as MRE11. This is a 3' to 5' exonuclease and also has endonuclease activity. We have NBS1, which is playing a role in damage recognition. We have RAD51, which is also known as a recombinase, and this is what's going to allow that step that's at the very bottom of the slide to occur for the genetic recombination to actually take place. Now on this slide you can also see BRCA2. Both BRCA1 and BRCA2 are proteins that have been associated with individuals that have breast cancer. Now, what I want to highlight, that when you hear about BRCA1 and BRCA2, they are in fact proteins that are involved in this homologous recombination mechanism of repair. In individuals that have breast cancer or have a family history of breast cancer with mutations in either one of those specific genes, have mutations that cause these proteins to not function properly, and hence those individuals have the potential to end up with cancer. So one of the things I always like to highlight is that when someone tells you they have the BRCA1 and the BRCA2 gene, everybody has the BRCA1 and the BRCA2 gene. It is specific alleles, mutant alleles, that are associated with breast cancer. Everyone has BRCA1 and BRCA2. But again, what I would like to highlight is that both of those play a role in regulating this activity, this homologous recombination to repair the double-stranded break. One of the last things that I'm going to talk about with this particular slide or lecture series is circling back to those histones. We talked quite a bit about histone structure and histone function when we discussed the organization of the genome inside a typical eukaryotic cell. As I mentioned then, there are a number of post-translational modifications that occur to histones to allow for the regulation of activity, either tightening up their association with the DNA or loosening up that association. So as you can imagine, for replication, you need to have that DNA association loosened up so all of those enzymes that we just finished talking about can play a role in getting that replication process completed. Same thing with transcription, and as you can imagine, with DNA repair. So what I want to highlight is that there has been some studies that have been done that have demonstrated one specific form of H2A called H2AX has been demonstrated to become phosphorylated in response to DNA damage. When it is phosphorylated, it plays a role in recruiting proteins known as cohesins. These are proteins that hold sister chromatids together. They also play a role, the phosphorylated histone also plays a role in recruiting chromatin remodeling complex. And chromatin remodeling complex is going to push nucleosomes away from that break so the enzymes that are going to repair the break can actually get to the DNA. So this mechanism of phosphorylating the histone in order to allow for the movement of the histone away from the break so that the damage can actually be repaired is thought to be playing a role in both mechanisms that we just talked about, both the non-homologous end joining as well as the homology-directed repair mechanism.